Okay, we're going to move on to proteins. And thing to know about proteins is just like all the rest of these molecules, shape and function, but guys, these are a little bit easier to identify because of their monomers, amino acids. These are the only monomers you will see both a carboxyl group, which is this guy here, and an NH2 group. Okay, so that's number one there. The carboxyl group is COOH. Again, the king point there is a carbon connected to an oxygen and an OH. And then the amine group, where you get the name amino acid, is the NH2 group here. Okay, so this is how you identify an amino acid: carboxyl, COOH, and amine group, NH2. Or depending on the chemistry, NH3, but typically NH2. The last part of this guy, and this is the most important part because this is what makes this amino acid what it is called the R group. Every amino acid has carboxyl and amine group. Each amino acid's R group is different. Why it's just kind of known as an R group. It doesn't say anything in particular about it. So each R group is going to be quite unique to each amino acid. And life in general, our bodies, plants, bacteria, your dog and cat at home, all use 20 amino acids to create life. Which, to think about it, is life is pretty complex. All the proteins that our bodies use, which we'll get to those, some of them, are all using just 20 amino acids, which is a pretty amazing feat that can do what it's do based on 20 amino acids. And all I want you to do here is I'm just linking to a Wikipedia amino acid or protein. I don't remember which one this is. Okay, it's linking to amino acid. So if you type in amino acid in Wikipedia, you'll get the same thing. You go down here and you see all these amino acids. Here's the 20 amino acids that life uses, to create life. This is what we use. All these amino acids make proteins. But again, in each one, and they're all arranged exactly the same way, or almost exactly the same way, where the NH2 group is over here to the left, and the carboxyl group is over here to the right, and the R group is on top. So this R group is very simple, just a carbon connected to some hydrogens and another carbon. Okay, so pretty small R group. Here's a larger R group here, connected to this amino acid, or part of this amino acid. Again, NH2 and the carboxyl group, amine group, NH2, and carboxyl group, COOH. This is arginine, and here's my amino acid joke. What's a pirate's favorite amino acid, right? Arginine. I know, that's pretty lame, but that's my amino acid joke. So, but guys, the point is, they all have an NH2 and a carboxyl COOH group. Okay, the rest of them, R groups, you don't have to know anything about them, just that they exist and they're all unique for each one. You can see pretty quickly on here that all these amino acids are unique because of their R groups. And let me go back really quick. This is what I was talking about earlier. Proteins are a great example of this. Proteins are literally just like a chain. Each link in the chain, the monomer in this protein is an amino acid, right here. Each link in this chain is an amino acid, and put together, all these amino acids form a protein. This is a perfect example of how a chain relates to the structure of a protein. And we have to get into the lipids next, I guess last, and an important part of the lipids are these fatty acids. Okay, these big long chains, kind of like tails, that are just bent lines. These are all just CH2 at each bend. Okay, just like in that propane was CH3, CH2, CH3, CH3 at the end, all the rest of them being CH2. So then this one has 18 carbons, not that you have to count it, but just an example for reasons you'll see in a little bit. We call this thing saturated because there are no double bonds, besides the carboxyl group. Okay, in this tail there are no double bonds, why it's called saturated. You can see it kind of spelled out here in a stick model on the bottom. Same thing, there are no double bonds besides the carboxyl group. In the tail there are no double bonds, saturated. Unsaturated means you take something out, taking out hydrogens. And if you take out hydrogens, it has to replace those bonds with a double bond. So a good example all the way down here at the bottom, we take out some hydrogens. Carbon has to satisfy its rule of 4 bonds, So it creates a double bond instead of having these hydrogens here taking up a bond. So this is called a trans configuration, but nothing you need to know. Just the reason that some of our foods are called trans fat, because they have this type of chain in it where it has this configuration, a double bond down here and just a straight long chain of CH2s basically. So to create a cis, which is the alternative form or configuration, the cis form, there's trans fat and here's a cis fat. Notice what happens in the cis fat. The hydrogens we take away to create that double bond are both on the same side, which causes this thing to bend. So instead of going horizontally across the page completely, it takes it takes an angle. It's roughly 30 degrees. Okay, now down to the right at about 4 o'clock or 4:30 if this were a clock. Continuing with these saturated, unsaturated, polyunsaturated. Okay, polyunsaturated just means more than one double bond. Okay, saturated is no double bonds in the chain. Unsaturated or monounsaturated is one double bond. Polyunsaturated, poly meaning many, polyunsaturated is many double bonds. And here's a good example. This is actually an omega-3 fatty acid. And hopefully you've heard of omega-3 before. In fish oil, things like that, oil, lipid, fat. Okay, here's an omega-3 fatty acid. The reason it's called omega-3 is because of its shape, and the 3 part comes from the third carbon from the end. Here's carbon 1, CH3 group. Here's a CH2 at carbon 2 that's not labeled. In carbon 3, that carbon is connected to carbon 4 by a double bond. Okay, so we have a double bond at carbon 3 from the end. That's why it's the 3 in omega-3 comes from. Here's a triglyceride. You're going to want to remember this structure. Okay, you will see this again. It starts with a glycerol molecule, and again, keep in mind this is a lipid, not a polymer. Okay, we can't add on to this thing to make it bigger. Here's what happens. This glycerol in the red rectangle is attached to these fatty acid chains, 3 of them, triglyceride, a glycerol with 3 fatty acids, triglyceride. That can be saturated, like this top one, no double bonds, unsaturated or monounsaturated, mono meaning 1, or polyunsaturated, more than 1, in this case 3. Okay, so connecting 3 of these fatty acids right here, the CO bond, and each one of these fatty acids connected to the glycerol. Okay, so this is a triglyceride. A glycerol, we call it the glycerol backbone. And then branching off from them are 3 fatty acids. The other lipid that we're going to talk about and quite a bit in this class, and this one may be the more important one, is the phospholipid. Okay, it just has this general shape, a phosphate head, which is this orange circle on top, of 2 fatty acid tails. And the one of them you notice here is bent. Okay, why is it bent? Well, the first one's saturated, right? No kinks in the chain. Besides, inside of here is back and forth, kink, back and forth, back and forth. But overall, it has a straight shape. Here's one double bond in the unsaturated, monounsaturated tail that causes it to take that bend at about 30 degrees. Here's the kind of zoomed in version of that. There you see the phosphate group, similar to what you saw in a nucleotide. A phosphate group is just a phosphorus with 4 oxygens connected to it. Here's that glycerol again, glycerol backbone, which is kind of like the base of this molecule, connected to those 2 fatty acid tails, and there you can see that double bond and how that one takes kind of that bent angle to it. The reason that these are more, or maybe the most important lipids, are because every single one of your cells, our cells, has phospholipids as the boundary in the membrane. And our organelles, which are kind of like the organs of the cell, many of them are also made up of membranes that consist of phospholipids. So to say these things are important for our bodies, for life in general, is pretty much the biggest understatement of this unit. Okay, these things are very important. So you're going to see a lot of this later on too when we talk about cells, cell membranes, and cell organelles. So don't forget this molecule. You're going to see it again. 2 things to point out: the hydrophilic and hydrophobic end. Again, just something to memorize now. It'll be more important functionally when we get to cells. Here's the table that is probably the table to remember for the function of all these macromolecules. Okay, you've already seen the structure of each one. Here's the function of each, and it's all summarized into one table. So I'm telling you right now, this table is pretty important. Here's the carbohydrates, first one we talked about. Basically an energy molecule. Okay, glucose, very important sugar we'll talk about quite a bit this year. Glycogen and starch, energy storage. Remember, glycogen energy storage for animals. Starch, energy storage for plants. You're hearing me repeat something, it's probably a good indication that it's important. And then structurally, we'll get into those too. You'll see carbohydrate sugars within DNA. Ribose for RNA, deoxyribose for DNA. That's why it's called DNA, because of that D right there in the sugar. Nucleic acids store genetic information and make proteins. Proteins have an unlimited amount. If the list would be a mile long. Proteins do almost everything your body needs done. Enzymes, very specific proteins that increase or start chemical reactions. Lipids, long-term energy storage, triglycerides, and structurally, phospholipids. Also functionally, but we're just going to say structurally here. We'll talk about the functions more of these when we get to membranes and cells. These links are very good demonstrations, animations of how a condensation reaction works, which you will need to know this for the test. So go to the PowerPoint that is at the website, or copy and paste, type into your browser these three websites, and this is going to show you exactly how these molecules form, something you're going to need to know. So click on each one of these and get the idea of how condensation reactions work. As a water molecule drops out, you'll see that very, very basic procedure done in these animations.